Hi students, in the last class we discussed about that meiosis. Within the meiosis 1 we discussed, right? Within the meiosis 1 we discussed about the prophase 1, right? Metaphase 1 and anaphase 1, telophase 1. Coming to the next one is the cytokinesis 1. The very next phase is cytokinesis 1. Cytokinesis 1 is nothing but division of cytoplasm. Division of cytoplasm. Whatever that cell organelles are there, the cell organelles are divided into two parts. So the step is called as cytokinesis process. First, suppose this is the one of the cell. The cell contains the two nucleus. The cell contains the two nucleus. See, the two nucleus contains the genetic material. The cell contains two nucleus. The two nucleus contains the genetic material. So we are seeing up to this portion in the last class, right? So within this class, that cell undergoes the cytokinesis process. The cell undergoes the cytokinesis process. So within cytokinesis process, see, one cell is divided into two cells. One cell is divided into two cells. Right? So this is about genetic material, right? This is about the genetic material. This is the DNA content, right? So how the cytoplasm division is occurred? How the cytoplasm division is occurred? We know within that animal cells, right? There is a plant cells are there. So plant cells cytokinesis is different from animal cells. Within animal cells, the method is called as cell furrow method. Within animal cells, the cytokinesis method is called as cell furrow method. Within plant cells, this method is called as phragmoplast method. This method is called as phragmoplast method. One is the cell furrow method, other one is the phragmoplast method. This one is also called cell plate method. This one is also called cell plate method. See, other names are also there here. Animal cell cytokinesis, also called centripetal method. Centripetal method. Animal cell cytokinesis process. Animal cell cytokinesis process is also called centripetal method. And remember, plant cells, the phragmoplast method is also called centrifugal method. Centrifugal method. So these are the other names for the cytokinesis within plant cells and animal cells. These are the other names for the cytokinesis within plant cells and the animal cells. So we are talking about the meiosis. Within that meiosis, okay, we are discussing about that cytokinesis 1. Okay, end of the cytokinesis 1, how many number of cells are produced? 2 cells are produced. So these cells are also called diad cells. These cells are also called diad cells. Diad indicates nothing but 2. So the stage is also called diad stage. The cells are also called diad cells. The cells are also called diad cells. So after that, cytokinesis 1, we completed meiosis 1. So up to this portion is meiosis 1. End of the meiosis 1, how many number of daughter cells are produced? 2 daughter cells are produced. End of the meiosis 1, how many number of daughter cells are produced? 2 daughter cells are produced. And next one is the meiosis 2. Next one, we will discuss about the meiosis 2. Within meiosis 2, we know again here there is an interkinesis phase is there. The phase is called as interkinesis. See, between the meiosis 1 and the meiosis 2, very short phase is there. This is very short phase. The short phase is called as interkinesis. See, generally in meiosis 1 there is an interphase is there. That interphase contains G1 phase, S phase and the G2 phase. But here meiosis 2, here interkinesis is there. Okay, this is not called as interphase 2 because here one of that DNA synthesis is absent. One of the DNA synthesis process is absent. So within the interkinesis phase, okay, interkinesis phase, G1 phase is present. G1 phase is present and G2 phase is present. G1 phase is there and G2 phase is there. Here which one is absent? Here which one is absent? Okay, S phase is absent. S phase is absent. Why here S phase is absent? Within meiosis process, only one time DNA replication takes place. Within meiosis process, only one time DNA replication takes place. The DNA replication also occurred within interphase. So within interphase, there is an S phase is there. But after the meiosis 1, there is an interkinesis phase is there. Within that interkinesis, the DNA synthesis is absent. That's the reason why this one is called as interkinesis. This is not called as interphase 2 because here DNA replication is not takes place. And remember, this is a very short phase. Right? That is about the interkinesis. Right after that interkinesis, again here prophase 2, metaphase 2, anaphase 2, telophase 2. Right? We'll discuss about that prophase 2. Right? Next one is that metaphase 2 and anaphase 2 and the telophase 2. So coming to the next one is the prophase 2. See, after that interkinesis phase, prophase 2, the next phase is the metaphase 2. See, every point is similar like mitosis, what we discussed. There is only one difference is there within that anaphase 2. See, the anaphase 1 is different from the anaphase 2. Remaining all points are similar. So here, prophase 2, within that prophase 2, this is the one of that, this is one of the cell membrane structure, right? See, this is the nuclear membrane structure. So condensation of chromosomes, that is nothing but whatever that chromatin is present. This is the chromatin. The chromatin starts the condensation. So the chromatin starts the condensation process, forms the chromosomes. Right? See, there is in condensation of chromosomes or condensation of the chromatin, right? Here forms the chromosomes. So if anybody want to write here condensation of chromosomes here, this is not exact suitable. Okay, just change it. The condensation of chromatin is the right word. So here condensation of chromatin, that chromatin involved in the condensation forms the chromosomes. So formation of chromosomes, condensation of chromatin, right? Decondensation of nuclear membrane. See, we already discussed whatever that nuclear membrane is there, the nuclear membrane, okay, disintegration, that is nothing but degradation process. So the nuclear membrane disintegrated, right? So nuclear membrane disappeared. Remember, the complete disappearance of nuclear membrane takes place within the metaphase. So here, decondensation of nuclear membrane, decondensation of nucleolus, and cell organelles also decondensed. Whatever that chloroplast, whatever that mitochondria, in case plant cell, chloroplast is there, right? Here mitochondria, whatever that endoplasmic reticulum, all cell organelles are okay decondensed. Decondensation of cell organelles, decondensation of nuclear membrane, decondensation of nucleolus, initiation of spindle operators. So this is one of the centrosome structure. Whatever that centrosome structure contains, centrioles are there. See, the centrioles are moving towards the opposite pole side. These are moving towards the opposite pole side. So once that movement takes place, once the centrioles are moved towards the opposite pole side, okay, which are involved in the spindle apparatus formation. So this is nothing but initiation of spindle apparatus. And coming to the next one is the metaphase 2. We know that metaphase 2, within that phase, meta indicates center. So here complete condensation of chromosomes will be takes place. So chromosomes are very clearly visible. Okay, we are doing here, there is an karyotyping process, means based on the chromosome size or morphology we are classifying the chromosomes. So that is nothing but the karyotyping. So within metaphase chromosomes are very clearly visible, and this is the best phase to study about the chromosomes within microscope. Yeah, by the help of microscope within cells all chromosomes are very clearly visible and very easy to identify, because within this stage all chromosomes are present at the cell center. So that point is also called equatorial plate. All chromosomes are arranged on the equatorial plate. So complete condensation of chromosomes is one of the point, right? All chromosomes are present on cell equatorial plate. So that's the reason why very easy to identify, right? All chromosomes are attached to the spindle operators. Here all chromosomes are attached to the spindle operators. So if the attachment takes place, then the separation of chromosomes will be takes place. Separation of chromosomes will be takes place, right? That is about the metaphase 2. So within metaphase 2, see I already said how the chromosomes are arranged at the center of the cell. So a group of proteins are there, that proteins are attacks the chromosomes to the center position. See the equatorial plate contains a group of proteins. That proteins attracts the chromosomes. So that by that way, within the metaphase all chromosomes are arranged at the center. Right? Next one we will discuss about the anaphase 2 and telophase 2. Coming to the next one is the anaphase 1. Here, anaphase 2. Right? Within anaphase 2, we know the process, but this is the only difference from the anaphase 1. Yeah, see what I am telling, the anaphase 1 is different from anaphase 2, because within anaphase 1 there is a reduction of chromosome number will be takes place, because there is no centromere division within anaphase 1. Whatever the homologous chromosomes are there, which are moved towards the opposite pole side. Simply there is no centromere division within anaphase 1. But here anaphase 2, there is a division of centromere will be takes place. So this is the one of the homologous chromosome, right? See, whatever the centromere is there, the centromere is divided. So this is one chromosome and this is another chromosome. So within anaphase 2, number of chromosomes are doubled. See, the same process is also takes place within that mitosis process. Within the mitosis, there is an anaphase is there. Within that anaphase also number of chromosomes are doubled. That's the reason why meiosis 2 is equals to mitosis. Meiosis 2 is equals to the mitosis, because here this one is also considered as equal division. Because the parental cells contains 2 chromosomes. Here the parental cells contains 2 chromosomes. See, the daughter cells also contains the 2 chromosomes. The parental cells contains the 2 chromosomes. The daughter cells, each daughter cell also contains the 2 chromosomes. So that's the reason why meiosis 2 is also equals to the mitosis process. That's the reason why meiosis 2 is considered as equal division, because here this is similar to the mitosis. Here whatever the chromosomes are there, the number of chromosomes are doubled. Initially this cell 2 chromosomes are present. This cell 2 chromosomes are present initially, right? Within that anaphase, after the division of centromere, here 4 chromosomes are present. Here also 4 chromosomes are present. So this is the increase in the chromosome number, or number of chromosomes are doubled. So here, how the number of chromosomes are doubled? Division of centromere. By the division of centromere, the number of chromosomes are doubled. Within meiosis 1, within anaphase 1, right, there is no division of centromere. No increase in the chromosome number. Then number of chromosomes are reduced to half. So we know that every chromosome movement from center to the pole side, say 30 ADPs are required. Here there is a pulling process. There is a chromosome pulling process by the energy. So the concept is here, whatever the chromosomes are there, the chromosomes are binds to the tubulin proteins, which are nothing but spindle operators. Then whatever that spin law operators are there, the spin law operators are by the utilization of energy, the energy in the form of ATP, the chromosomes are pulled. The chromosomes are pulled towards the pole side. So once the chromosomes are reaches the pole side, right? See the next phase. The phase is the next phase. So within that anaphase 1, okay, number of chromosomes are doubled. Division of centromeres occurs. Here we know the point is 30 ATPs are required for the movement of one chromosome from center to the pole side. See, in this phase, chromosomes are moving towards the pole side. And coming to the telophase 2, within this phase, here again two cells are there. This is the reverse prophase. So reverse prophase is nothing but opposite to the prophase. Here decondensation of chromosomes. Here chromosomes are present, right? Here decondensation of chromosomes. That is nothing but formation of chromatin. And condensation of nuclear membrane. So here nuclear membrane is absent. So here nuclear membrane is appeared, right? Nuclear membrane formation takes place. Nucleolus formation takes place. And cell organelles formation also takes place. That is nothing but condensation of nuclear membrane, condensation of nucleolus, and condensation of cell organelles, right? So that is about the telophase 2. Last one is the cytokinesis 2. We'll discuss about the cytokinesis 2. So this is the last phase within the meiosis 2, cytokinesis 2. So we know that cytokinesis process. See, there is an animal cell furrow method is there, plant cell phragmoplast method is there. That same manner, centripetal method within the animal cells, and centrifugal methods within the plant cells, right? So I already said animal cell cytokinesis requires less time compared with that and plant cell cytokinesis process, because within the plant cells, the cytokinesis, see, there is a cell wall formation is also required, but within the animal cells there is no cell wall. So less time is required for the animal cell cytokinesis, more time is required for the plant cell cytokinesis. So remember, see, this is the cytokinesis process. Division of cytoplasm takes place. So end of the meiosis 2, 4 daughter cells are produced. Remember, these 4 daughter cells, the state is the haploid state. Because initially we are taken 4. The cell contains number of chromosomes are 4. The number of chromosomes are 4. The cell contains number of chromosomes are 4. But within the daughter cells, only 2 chromosomes are present. So within the meiosis process, the number of chromosomes are reduced to half. Within the meiosis process, the number of chromosomes are reduced to half. See, significance of meiosis. See, this is a very important one. The significance of meiosis is very important one, because it's maintain the constant number of chromosomes. Constant number of chromosomes in every species. Those the cells which are involved in the meiosis process, all these, whatever the species which are involved in the meiosis, so all the species, the number of chromosomes are maintained constantly. Means human species, number of chromosomes are 46. So different species, we already know that there is an order of chromosomes, we already discussed. So every species having a constant number of chromosomes. That constant number of chromosomes are maintained by the meiosis, because whatever that the gametes are produced, the gametes having that haploid set of chromosomes, once the fusion takes place, then again the body state is the diploid. Again, new organism, new gametes production, right? Again, new organism. So continuously in the every species, constant number of chromosomes are maintained by the meiosis process. The next point is evolution and adaptation. Evolution and the adaptation. See what I am telling here. Meiosis is the very important one for the generation of the new species, because humans also evolved like that. How humans are evolved from the bacteria, or how humans are evolved from the chimpanzees by the crossing over process. Within that meiosis process, there is a crossing over is occurred. That crossing over supporting for the generation of the new characters. That new characters are nothing but evolution and the adaptation. So this is about that meiosis. So this is about the cell cycles and cell division chapter. Next class we will discuss about the biomolecules chapter.